Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the Deep Dive, the show where we grab your source material and distill the key nuggets of knowledge you really need to be well-informed.
Today, we're undertaking a super focused deep dive tailored specifically for those of you mastering foundational concepts.
That's right. We're drawing heavily on NCERT Class 11 Biology, Chapter 9, Biomolecules, with an eye toward that rapid and thorough learning you need for complex exams like the UPSC.
And, before we unpack the molecules of life, we have a quick but really vital request for our learners.
Go ahead.
If you are watching this on the Simplified UPSC by PPP YouTube channel, please do take a moment right now to hit that like button.
It really helps.
It does. And share your valuable comments, and most importantly, subscribe. This content is possible thanks to the fantastic support of our existing paid members.
A huge shout out to all of you.
Yes, a huge shout out. Your membership helps us keep creating these super useful, high-quality deep dives. And if you are capable, please consider joining the paid membership to support us further.
Okay, let's unpack this. We are diving into the very chemical structure of life itself, the biomolecules. The sources show us how biologists separate the stuff of life chemically.
Yeah, using these things called the acid-soluble and acid-insoluble pools.
Right.
And, that process is just fundamental. We need to categorize these chemical constituents. What's fascinating here is that every living tissue, whether it's plant, animal, or microbial, everything, it contains both organic and inorganic substances. We are literally breaking down the chemical architecture of the cell.
So we've identified some inorganic heavy hitters right off the bat, things like phosphate and sulfate.
That's SO43 and SO42, yeah.
These, along with the other minerals, they sort of set the stage. But where does the line actually fall between the small molecules and the large structural components?
That's the key question, and the differentiation relies heavily on molecular weight. This often forms the basis of direct conceptual questions on exams.
So what's the dividing line?
Well, for micromolecules, the ones in that acid-soluble pool, these compounds typically have molecular weights ranging from about 18 to around 800 Daltons.
Okay, so these are the basic building blocks.
Exactly. Your amino acids, simple sugars, the small stuff.
And then you have the macromolecules, the acid-insoluble fraction.
These are the giants. We're talking molecular weights of 10,000 Daltons and above. This includes your proteins, nucleic acids, and polysaccharides.
Okay, but there's a really important exception here, a detail that I think is crucial for any learner: lipids.
Ah, yes, a lipid exception.
They're found in the acid-insoluble fraction, even though their molecular weight is actually pretty low, right?
Yeah.
Generally less than 800 Daltons.
It's a critical detail, and it's not about the size of the molecule itself, but about its context, its association with membranes.
I see.
When you analyze the tissue, the lipids are part of these membranes, which break up into little vesicles. And those vesicles are large enough that they get separated out with the true macromolecules.
So it's a procedural artifact, not a chemical one.
Precise.
So what does this all mean for the overall composition of a living organism when you look at the big picture?
It means water absolutely dominates. Water is overwhelmingly the most abundant chemical in living organisms.
Why a lot?
Oh, by a huge margin. And after water, the remaining composition is a mix of these organic and inorganic constituents, with proteins usually taking the biggest share of the dry weight.
Okay, let's focus on the organic micromolecules then, the primary ingredients. First up, amino acids.
What's fascinating here is their precise structure. They're called alpha amino acids.
Meaning they're substituted methanes.
Exactly. They contain an amino group and an acidic group, a carboxyl group, attached to the very same carbon, the alpha carbon.
And the R group is the part that changes.
That's the variable part, and it's what determines the identity of the amino acid. It's amazing to think that only 20 types of these occur in all proteins.
Just 20. And then we have the components of our genetic material.
Right, the nucleotide bases. And here we make another key distinction.
Purines versus pyrimidines.
Yep. Purines like adenine and pyrimidines like uracil, which is a specific example mentioned in the sources. This distinction is what helps define the whole larger structure of nucleic acids.
Now, moving beyond these standard molecules, the source introduced a secondary classification of metabolites. Why is this distinction so important for studying biology?
It's a really interesting classification. You have your primary metabolites.
Which are the common building blocks we've been talking about. Amino acids, sugars, things you find in basically all animal tissues.
Right. They have identifiable functions in normal physiological processes, but then you have the secondary metabolites.
And these are thousands of other compounds.
Thousands. Found specifically in plant, fungal, and microbial cells. We're talking alkaloids, flavonoids, rubber, essential oils, even antibiotics.
And they perform functions unique to those organisms.
Exactly. Often for defense or attraction. They're not directly involved in growth, but they're crucial for survival in an ecological context.
Okay, so here's where it gets really interesting, I think. Let's talk about proteins, the ultimate heteropolymers.
Yes. They're linear chains of amino acids, all linked by peptide bonds. But their structure is, well, it's way more complex than just a simple chain.
So much more.
That's right. Biologists describe protein structure at 4 hierarchical levels. You have to move from the linear sequence to this complex 3D shape.
So level 1 is the primary structure.
The primary structure, yeah. It's just the simple sequence and positional information of amino acids, which one is first, second, and so on.
But that simple sequence dictates everything else.
Everything. From there, you get the secondary structure, which is all about local folding patterns.
Like alpha helices.
Alpha helices, beta sheets, yep.
Yeah.
And then you get the tertiary structure.
And that's the overall 3D folding and shape, the functional form.
Adds the money shot, yes. And finally, you have the quaternary structure.
Which is how multiple protein subunits come together to work as a team.
The organization of multiple protein chains? Think of hemoglobin.
Right. But proteins aren't just structural. Many of them function as enzymes. And this is critical because our sources state there is, and I'm quoting here, no uncatalyzed metabolic conversion in living systems.
That is an incredible statement, isn't it? It just raises this important question. How do they actually work?
How do they speed things up so dramatically?
Well, enzymes are catalytic proteins. They convert a substrate, which we call S, into a product, P, with extreme efficiency.
And the entire process hinges on the active site.
The active site, yes. It binds the substrate, and this forms an enzyme-substrate complex, or ES.
And this complex is very short-lived.
Very. It quickly leads to an enzyme product complex, or EP, and then finally it releases the product and the unchanged enzyme.
So the enzyme is ready to go again.
Exactly. And crucially, the enzyme works by lowering the energy barrier, the activation energy required for the reaction to happen.
It digs a tunnel through the mountain, so to speak.
That's a perfect analogy. It makes the reaction millions of times more likely to occur.
And their function is highly, highly sensitive to the environment.
Precisely. Because it all depends on that 3D tertiary structure. Enzyme activity is regulated by external factors that can alter that shape.
Like temperature.
Temperature, pH.
A change in the substrate concentration.
Right. Or the binding of specific chemical regulators. Any of these can turn an enzyme on or off.
Before we transition, we have to mention 2 facts that just make proteins truly monumental in biology. I think these are vital memory points for any aspiring learner.
Absolutely. Go for it.
First, the most abundant protein in the entire animal world is collagen.
Our structural framework.
And second, the most abundant protein in the whole biosphere is Rubisco.
Ribulose bisphosphate carboxylase oxygenase, the engine of photosynthesis. Knowing those 2 is just non-negotiable.
Couldn't agree more.
So let's connect these building blocks back into the overarching story of life metabolism. Metabolic pathways. They're classified into 2 opposing forces.
Anabolic and catabolic.
Right. Anabolic pathways are the biosynthetic ones. They consume energy to build complex structures from simpler ones.
For example, assembling a protein from amino acids.
Or converting acetic acid into cholesterol. That's anabolism. It's construction.
And then there's catabolic pathways. Degradation.
These are the ones that release energy by breaking down complex structures. The classic example everyone learns is glycolysis, that 10-step degradation of glucose to lactic acid.
And the currency driving all of this is bond energy.
Yes, and the most important energy currency in the cell is stored in a molecule called adenosine triphosphate, ATP.
ATP.
That stored bond energy is what living systems trap and then utilize for absolutely all work, whether it's biosynthetic, osmotic, or mechanical. It's the universal fuel.
Okay, finally, let's quickly look at the blueprint itself.
Yeah.
The nucleic acids, DNA, and RNA. What are the essential structural details we need to remember?
Well, they're polymers made of nucleotides, and they serve as the genetic material. And the classic example, of course, is DNA, which has that famous Watson-Crick double helix model.
2 strands running anti-parallel.
Like a divided highway, exactly. And the backbone is formed by these alternating sugar phosphate chains.
giving it strength.
Right. And the nitrogen bases, they project inward, facing each other, perpendicular to that backbone.
And the base pairing is strict and compulsory.
It has to be. Adenine, or A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. This ensures the helix has a uniform width and, more importantly, allows for perfect replication.
We've covered the fundamental chemical organization of life, from simple amino acids all the way to the complex mechanics of enzyme catalysis, all synthesized directly from this foundational material.
And for the dedicated learner, remembering these precise distinctions, like why lipids end up with the macromolecules, or the difference between primary and secondary metabolites, and those defining proteins like collagen and Rubisco, these are the details that ensure a really deep understanding of cellular processes.
This has been a truly concentrated deep dive into the essence of biomolecules. And here's a final provocative thought for you to explore on your own.
I'm listening.
If enzyme activity is so sensitive to changes in temperature and pH, how do organisms like archaebacteria survive in extreme environments, like hot springs or deep oceans? Think about what adaptations their protein structure must possess.
A fantastic question. What makes their proteins so robust?
Something to think about.
And once again, to all our listeners preparing for exams and seeking deeper knowledge, we really hope this deep dive was useful.
Please support our efforts on the Simplified UPSC by PPP YouTube channel by liking this video, sharing your comments, and subscribing.
And a huge thank you again to our existing members whose support makes this essential content possible. If you can, please consider joining our paid membership. Until next time, keep learning.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.